All right, good morning, everybody. Thanks for joining us. My name is Mick. I'm from Biognosis. I'm from the marketing team. I'd like to first start the seminar by giving a quick introduction to our software products, and then we'll hear from Ikai about multiplexed DIA analysis using Spectronaut for phosphoproteomics of extracellular vesicles. Then we'll hear from Birgit to discuss quantitative DIA for bone tissue and PTM analysis. All right, so at Biognosis we have three products. I think most people are familiar with Spectronaut. It's our flagship product for analysis of DIA data, both library-based and library-free. We also have SpectroDive for targeted proteomics analysis. And then we also have SpectroMine, which supports analysis of DDA data. So very briefly, here's Spectronaut. It's our leading flagship software. It's one of the best performing DIA products available. We'd like to really discuss and promote the library-free aspect. We think it's very powerful, but it's more than just a little louder. It's more than just a search engine. It also has customized visualizations and a very full-fledged ability to adjust and customize reporting. So last ASMS we released Spectronaut 19, and I just want to highlight some of the performance improvements. You can see that we don't just look at a few data sets that may make the product look very good. We have looked at 130 different data sets from different mass spec vendors, and you can see on average we're looking at about 10% performance improvement over Spectronaut 19. But we're not just looking at IDs. We also see improvements in what we call candidate discovery. These are identified proteins that we discover that are differentially abundant, so we can actually see from a quantitative perspective an improvement in the number of proteins that are changing. Oh, sorry, I meant to say we see about 11% improvement in this newest release. But we're not only working on performance in the idea of improving the numbers that we see, but also improving our computer utilization. As the data size is growing, we know that we have to be able to analyze it faster. And you can see we see about a 90% reduction in the amount of memory that we need to use for large-scale analysis. And with that, I'd like to introduce our first speaker, Ikai Liu from Purdue.
Hello everyone. Can you hear me? Yep. Okay, good morning. So my name is Ikai Liu. So I'm from Dr. Andy Tao's group at Purdue University. And today I'm very honored to present this work on behalf of Dr. Tao. So in this presentation, I will introduce our recent study using multiplexed DIA method to study the EV proteomes and the EV phosphoproteomes. So in this presentation, I will mainly focus on the proteome results, and I will share some preliminary data on the phosphoproteomes. So let's start from the EVs. So extracellular vesicles, EVs, are small particles secreted by almost all types of cells. And they, in the biological systems, they are an important mediator for cell-to-cell communications. That means they can carry biomolecules like the proteins, PTMs, and the RNAs, also metabolites from the donor cells to the recipient cells. And because they can carry this informative information from the cells, and also this EV membrane can protect these cargo molecules from degradations. So they are like a promising source of the targets for detections. And also, most importantly, these EVs can be secreted into the biofluids like the plasma and the urine, so they are further promising source for the non-invasive biomarker detections. So here is the general workflow for EV proteomics detection. So the EVs can be isolated from the several sources, including the cell culture media, tissues, and the biofluid is like a blood and the urine. And later we need to do a step called EV isolation. So this step, typically we use ultracentrifugation or other method like affinity-based method to remove the contaminants from the samples and the enriched EVs from the samples. And after that, the enriched EV can be further subject to the sample prep for the EV proteomics. And then after that, we can identify some significant targets for the potential disease biomarker and to be further validated. So in the past 10 years, our group have been working on developing different techniques for the EV proteomics and the phosphoproteomics, and we also apply these techniques in detecting the EV proteins biomarkers for different types of cancers. So one of the important challenges for this EV proteomics is the low abundance of EV-derived proteins compared to the cellular proteins or the free circulating proteins. So if the proteins from the cells or from the free circulating protein like plasma proteins like albumin or other free proteins, their high abundance can overshadow the mass spec signal of the EV proteins. So in this case, we need a more sensitive method and provide a better quantification and also increase the throughput. So in this study, we try to apply the recently introduced method called multiplexing DIA. So this method combines the advantages of both methods, which is multiplexing, which provide the sample pooling and provide less variation between the samples, and also the DIA method, which provide better sensitivity and also better quantification. However, the major challenge of this method is that it further increases the complexity of the DIA spectra. So this might be the major limitations. However, recently there are several groups have been using this technique to further prove that this technique can provide a better sensitivity and also better proteome coverage. For example, the PAX DIA developed by Dr. Slabov's group, and also the Dr. Liu's group used this technique to combine the p-Silac with the DIA method. And also most recently, Dr. Mann's group also used this multiplexed DIA based on the dimethyl labeling, and they detect the single cell proteome and prove that the sensitivity is much better. So in this study, we're curious, like there are several factors we know is well known to affect the DIA outcomes. So for example, the spectral library can influence the DIA search results, and also the software tools, different software tools might also provide different outcomes for the DIA results. But there's still not a very clear evaluation of these factors on the multiplexing DIA. So in this study, we try to explore these different factors on the multiplexing DIA using the EV samples. So our objective is to provide optimal multiplexing pipelines for quantitative proteomic analysis of EV samples. And later I will show an example of using the optimal pipeline to study the cancer. So let's go to our workflow. So first, we need to do a sample preparation, which is EV preparations. So in this study, we use the cell model systems. So we isolate the EV from the cell culture media, which is, so we isolate the cell culture, isolate EV from the cell culture medium, and using this EV trap technique. So this is a technique developed by our group. And so this is the functionalized magnetic beads. So it has affinity to the AV structure. So we have been using this technique in the many studies and prove this high recovery of the EVs using these techniques. And after that, we isolate the EV on the beads, and we directly perform the on-bead SP3-based sample preparations. So we have been proven this workflow in the previous studies, and it can provide an efficient recovery of EV proteins and also make the whole sample preparation more efficient. And after that, we got the digested peptides, and we label them with 3-channel dimethyl labeling. And then after that, we pour the sample together into one sample. So next, we want to optimize the mass spec acquisition method, and at the same time, we want to build a library for the DIA search. So in this case, we have the proteome sample, pool samples, and after that, for the phosphoproteome, we do another phospho enrichment to get the phosphopeptide samples. And in this case, we hypothesize the EV proteome is less complicated than the cell samples. So we assume that we can use just the tip-based fractionation to achieve the enough quality to separate the complex, the EV proteins in the samples. So in this case, we use just normal stage tip-based basic pH fractionation and get the 8 fractions eluted in different ACN concentrations. And by injecting this 8 fraction into a timsTOF HT platform, and we perform the DDA passive method to acquire this 8 fractions library. So this library, we will use this library for the library-based DIA search. And moreover, this library can be used for our DIA method optimization. So in here, we use the software called PYDIAD, which developed by Dr. Mann's group. So we use this software to further optimize the timsTOF acquisition windows. So in the output, we can know, like we can detect our precursor distribution in the timsTOF, on the timsTOF, and we can set up the windows based on this precursor range. And then we can get the optimized window for the DIA acquisition. So in the end, we perform different pipelines for the DIA search. So we have the proteome sample and the phosphoproteome samples. For the individual run, we run the DDA passive mode, and for the DDA data, we use the FRAC pipe to analyze the data. And for the DIA passive, we run it with library and also without library, and search their data using the Spectrona and also Diane. So we want to evaluate these different pipelines to determine the optimal results from these pipelines. So first, I want to quickly show the EV characterization. So typically, if we want to do the EV study, we want to fully characterize the EV samples to make sure that our EV sample is not to have too many contaminants, or the sample is not clean. So in this case, we validated using the Western blot to detect several EV markers and also the negative marker connexin. So these markers should not exist in the EV samples, but in the cell samples. And also we use a nanoparticle tracking analysis and also TEM to evaluate the particle distribution and also morphology. So after we get the EV samples, then we perform different pipeline on these EV samples. So there are 5 conditions we want to evaluate. So the first one is the normal DDA method, and the second one is the direct DIA method using Spectrona, and the third method is the library-based DIA using our fraction library and the search with the Spectrona. And the fourth condition, we use the same fraction library, but using Diane to do a search. And then the last one, we use the predicted library from the previous studies and use Diane to do a search. So in the identification result, we can see that the DIA method could significantly improve the ID compared to the DDA method. And among the DIA method with the fraction library, the ID further improved compared to the library-free method and also predicted library. So later we further evaluate the specificity of the method toward the EV proteins. So we took the top 100 EV proteins from the existing EV database, ExoCarta. So there are the database, at least the EV proteins that are commonly identified across different studies. So we choose these top 100 EV proteins and see how many top 100 proteins be identified in different pipelines. So we can see that the library-based DIA using Spectrona identified the highest number of the top 100 EV proteins. And when we look detailed into the specific proteins, we can see that the most common protein, for example, CD9, only identify in the these 3 DIA workflow, but not the DDA and also predicted library workflow. So which means the library, the fraction library provides the specificity of the detection. And then we further evaluate the quantification of these pipelines. So we mix the different labeled peptides in the different ratio, which is 1 to 2 to 4. Then we do also the single run of the samples and with the different pipelines. And in summary, we found out that the library DIA using spectronaut search provide a better performance of a quantification compared to other DIA methods, but it has a comparable quantification results compared to the DDA. So we also do the phosphoproteomics on this using these different pipelines, and we are still collecting the data. So this is kind of preliminary data for now, but we can clearly see that with the library DIA and also using other DIA methods can provide more class 1 phosphopeptide compared to the normal DDA methods. And also, furthermore, with the library, it increased the ID about 1,000 class 1 phosphopeptide be identified. And it's also interesting to see that if without library using direct DIA method, it provides a more non-phospholipeptide be identified. Means the selectivity of the phospholipeptide is lower compared to the library-based DIA. So which means in terms of the phosphoproteomics, we think that the library is still necessary to provide the identification of the class 1 phospholipeptide. So here I want to show the example of using this multiplexing DIA pipeline to study the EV proteins. So here we choose these cancer cells models. So this is the intrahepatic cholangiocarcinoma cells, which is iCCA cells. And this iCCA is a cancer, it's a bile duct cancer, and it's very aggressive. And currently there are many subtypes identified. So one of the common subtypes is this IDH1 mutant subtype. And the IDH1 stands for isocitrate dehydrogenase 1. So these subtypes usually show a poor prognosis, and also sometimes they show the drug resistancy. So there's a need to identify this subtype and also provide a better treatment outcome. So here the IDH1 played an important role in converting the isocitrate to alpha Kg. So if this IDH1 mutation happens, this alpha Kg can further convert it into an unco-metabolized D2HG. So this D2HG accumulation can further change the gene expression and provide the cancer phenotype. So we want to see so there are many studies have been proven the proteome change in the cells level, but we want to see if this cells level change can be also reflected into the EV levels, which means we can use the EV as a potential source for detection of this subtype and also provide a treatment monitor. Oh yeah, so actually there's a proof inhibitor currently. So one of the inhibitor we are going to use is this AG120. So this is a known inhibitor for the mutant IDH1. So in this experiment, we use the 3 cell models and these 3 conditions, including the CCLP cells, which is the IDH1 wild type iCCA cell line, and also the IDH1 mutant cell line IRBE, and also the mutant cell line treated with the inhibitor. And then we took the cells and also EVs and label them separately and also detect them using the library-based multiplexing DIA, the workflow we just established before. So there's one thing I want to mention, that we found out that the cell samples, if we use this multiplexing DIA workflow for cell samples, it provides the less ID compared to using the predicted library. So we think that the tip-based fractionation here does not provide enough resolution to to provide a better spectrum library for the cell samples. So we think that for the cell samples, in terms of the multiplexing DIA, you need to do the deeper fractionations or using the predictive library, which provide better identification compared to our tip-based fractionation. And then we further compare our samples. So we first compare the IDH1 wild type and IDH1 mutants. So these two samples, we found there are a large overlap between the identifications. And then later we do the correlation between the cell result with the EV result. We found a lot of proteins be overlap between these two results. And also there are many significant proteins be overlap in these two results. And there are many non-IDH1 mutation-related proteins, for example, the CBR1 and also S186, and also LDH1A1. So some of these proteins are non-IDH1 mutation-related proteins. And furthermore, we took these upregulated proteins to do a gene ontology search, and we can see that there are many pathways related to the IDH1 being enriched. For example, this pentose phosphate pathway and also fructose metabolism. So since IDH1 involved in the TCA cycles. So this data support that this IDH1 mutation associated the proteome change in cells can potentially reflect in EVs and be potentially used for the future disease diagnosis. And I'll quickly go through this inhibitor treatment data. So the inhibitor treatment, in terms of the inhibitor treatment, we didn't find a good overlap between the cell results and the EV results, assuming that the cells response may not be reflected into the EVs, or the reflection may not happen in the proteome level. So, but we still took the EV result to do to identify some significant proteins, and some of the proteins actually related to the IDH1 mutations, but their function in EV was still unknown. And furthermore, we took the downregulated EV proteins to do a gene ontology search, and indeed we see some pathways, for example, for RNA capping pathways. So this is also known pathway related to the IDH1 mutation. So potentially this EV may carry this information from the cells, but further validation is still needed. And we here provide some example proteins, EV proteins, that can show the response in the condition between the IDH1 mutation and also inhibitor treatment. Hopefully, in the future, this potential target can be used for the IDH1 mutant subtype differentiation and or the inhibitor treatment for a non-invasive biomarker. So in the conclusion, I hope today I show the data to show you that we kind of evaluate the optimal dimethyl labeling-based multiplexing DIM method for quantitative detection of EV proteomes and phosphoproteomes, although we are still collecting phosphoproteome data. And in conclusion, we found out the fraction from the simple tip-based fractionation provide enough resolution for the EV samples, but not the cell samples. And we also first demonstrated that the proteome change in for the IDH1 mutation and the inhibitor treatment can be detected in the EVs. So further validation is still needed. So in the end, I want to thank Dr. Andy Taus for supporting this project and my PhD work, and also my colleagues and my undergrads help for this project. And most importantly, I want to thank the people from Biognosis, Monica and Rachel, for helping us to set up the workflow. And also, we have been using Spectronaut for 5 more years, so we like using it. And I hope today's presentation can give you some information. Thank you so much.
Thanks. Excellent presentation, Ikai. So I'd like to hold any questions for the end of the session. And with that, I'd like to welcome our second speaker, Birgit.
Thank you so much. Thank you. Well, good morning. Thank you. Can you hear me okay? All right. Thank you, Mick, and thank you to Biognosis. We've been using Biognosis for many years now, and It really has helped us in all of our workflows. And today I will give you some insights how we're using some of these more novel features for PTM analysis. My lab has been doing post-translational modifications for a really long time, and it actually goes along well with the first speaker. A lot of the functionality in cells is dynamically regulated by post-translational modifications. So here you can see a photo of the Buck Institute. We're in Northern California, just at the north end of the San Francisco Bay in California. So my lab, we actually indeed do a lot of different things. This is like a little sketch that shows you the variety of research projects we have. They all are age-related. We are at a research institute for aging, but a lot of age-related diseases have actually aging as risk factor, and so this is very evident for neurodegenerative diseases, for osteoarthritis. Indeed, really cancer has age as high risk factor, and indeed in some projects, particularly my scientist Rana, she looks at healthy aging. So we're really interested in keeping people aging healthy and increasing the health span versus the survival span. And when does healthy aging become detrimental and maybe make it more prone for an age-related disease? So these are really fascinating questions, and today I'd like to talk to you a little bit about kidney diseases. It's actually very underappreciated how many aged people have problems with their kidneys, and it really can implement and indicate a lot of detriment in quality of life. And the same is actually true for osteoarthritis. Many people, as they become older, have mobility problems, and really problems with bone and musculoskeletal systems can really hurt a lot, and this is what really can impact quality of life. But we're talking today about post-translational modifications in the context of those two projects, and I just have one general slide about PTMs. So PTMs are really very special. They can actually be very dynamic. They can go on and off super quickly. Oops, I don't know why this is moving, sorry. But at the same time, you can have neutral loss from these modifications. They can have low site occupancy, and often we use PTM enrichment strategies. So this all influences also, of course, the workflows, how we analyze the data. And indeed, it's really important to be really sure about the quantification of each PTM peptide, because other than in protein quantification, you may only have one peptide that really now gives you the PTM status of that particular cell. So PTM workflows are actually quite different from other lysate or whole protein workflows. So they come with challenges and opportunities, I always say. So let me just show you one other slide indicating here that maybe you can see this is supposed to be a little mitochondrial sketch. And so each protein may actually have multiple different PTM sites. So you may have some proteins that have multiple PTMs on the one protein, and each of the peptides is an indicator for different PTM, for example. And they all may be regulated very differently. So it's not a protein level quantification. It has to really be quantification on the peptide level on each specific peptide that contains post-translational modification. And sometimes you may have a multiplicity where one peptide can have 3 or 2 PTMs. So then the whole quantification gets also complicated, and Spectronaut has really been able to kind of decipher these things. So this, for example, shows you that maybe in one protein here called HMGCS2, which is a metabolic protein, you have different sites. And each of those are, in this case, succinylated or acetylated. and then the color code shows you the regulation, the level of upregulation when you compare different conditions. And it can be that some site is super highly regulated and the other site much less regulated. And actually, I really love to do dynamic studies with post-translational modifications because often you see trajectories. Some sites you see a really super strong increase of maybe succinylation, maybe starting very early and then going up and up and up. Others you have maybe a more slower start of regulation, and some PTM peptides may actually plateau. So these are all down the road analytical challenges for a search engine. So this is really, this first little sketch here is supposed to show you the fold regulation that you can determine in PTM peptides. But actually, for some biologists, more importantly than that, is what we call stoichiometry. So that's a site occupancy. And a lot of biologists are actually more interested in stoichiometry versus in fold regulation. I mean, you may have a regulation of 200% where it goes up twofold, or you may have a regulation where it goes up tenfold. But maybe the site occupancy is only 0.1%. So a biologist may be more interested in a PTM site where the occupancy is maybe 20%. So meaning 20% of this unmodified peptide now gets regulated and, say, phosphorylated or succinylated. So that is another dimension of looking at PTMs. And Spectronaut has now features where you can calculate these stoichiometries. We've done this before with in-house workflows, but it can become very complicated. So having this as part of Spectronaut is really, for us, really important and really, as I said, really important for the biologists down the road. Because why are we interested in the whole thing? Well, what is really important is how does the PTM portfolio change function, how does it change protein-protein networks, and what pathways are affected. And, often these PTMs are somewhere in the 3D structure of a protein, and it's actually really interesting to see which PTM sites are hotspots. Is this maybe near a functional domain? Sometimes you have PTMs that are near the site of where a certain substrate of a protein that may be a metabolic substrate gets bound. So those sites are particularly important. And so we often actually look at the 3D structure of proteins where we see phosphorylation or succinylation, PTMs in general. So, PTMs are really complicated and complex, but also relevant. So that's why we like those. So Spectronaut has these stoichiometry workflows now, and for us this has become really important and is included in all the reports, and you can customize the reports. I will show you in one of the next few slides. So today I wanted to show you two different ways how you can use Spectronaut for PTM work. In fact, the first one, this is some nomenclature that Joanna came up with. We call it a dual enrichment, where you do basically enrichment for PTMs. So when you do that, you have to look at the PTM enriched samples. This is what we usually would do. But if you want to also have insights into stoichiometry, or if you maybe want to do normalization for protein level changes, you also have to look at the lysates, right, where it's the sample before enrichment. So typically we take like 2% of the sample and just keep it non-enriched, where we just look at the protein level changes. And so basically you do two acquisitions, one for the enriched PTMs and one just whole lysate for protein level changes. But then, and this is really the reality for some PTMs where they have very low, let's say, stoichiometry, where you have to enrich. But there are some, modifications that are so abundant, you don't even have to enrich. So we call this a single workflow. You can take the acquisition, you get your PTMs from that, and you get your protein level quantification also. So I will show you actually examples for both workflows where you do enrichment and where you don't have to do enrichment. So the setup in Spectronaut is just slightly separate for that. That's why I'm showing you both. So the first project that I would like to show you is a supplement of a diet in a kidney injury project in mice. And basically you take this diet, I call it DC8 here, it's a dicarboxylic acid, it's a natural product, and then mice with a normal diet, and then you have kidney injury or no. You do the lysis of the tissue, and then as I mentioned earlier, one arm you actually do the enrichment, and the other arm maybe with 2%, you just do the lysate digestion and just look at this for the protein level analysis, and the upper part is for the PTM level analysis. And then you can use Spectronaut if you want to do protein normalization. That is really for, when you do see an increase in PTM, some people ask, well, was it because the site was regulated, or was it because the protein was upregulated? So Spectronaut allows for either doing that normalization for PTMs or not. We have until now usually done this normalization, but I've had a lot of questions, discussions with Martin Larsen, for example, because in the end for the cell it doesn't matter where the regulation comes from, whether it's protein level or PTM level, the cell sees the PTM go up. But sometimes it's just kind of nice to know, is it PTM regulation or protein regulation? So when you do the normalization, you can decipher really the PTM regulation. So we do that a lot, actually. And then the other example I show you will be about some bone proteomics. Well, you might be surprised, there's thousands of proteins in bone matrix. It's a calcified matrix, but there's a lot of proteins in there. So I'll show you an example of ApoE mice, ApoE2, E3, E4, you may know that that's a risk factor for Alzheimer's. And we're looking at hydroxyproline modification, which makes the crosslinks of collagens very important for bone quality and bone strength. So that is so abundant, we don't have to do an enrichment. So we can do everything from the non-enriched sample. So I just want you to appreciate this different workflow where here you just take the sample and acquire it, versus in the upper part we do the enrichment and then we do the protein digestion from the lysate also. But in the end you use Spectronaut for both stoichiometry and normalization, whether you use enrichment or not. So let's start with this first project in the kidney injury. This is together with my collaborators in Pittsburgh, Sunny Sims-Lucas and Eric Götzmann. So we have found that, oh sorry, I don't know why this is doing this. So we have found that this natural product can really protect against kidney injury. And so basically this shows you on the left, you may have some mice. Oh, it's doing some automatic moving forward. Oh my God, so let's see. So you can take the mice, you can induce kidney injury. Indeed, you have 2 kidneys in each body, so you could actually induce injury in one kidney for the mouse where you just stop the blood flow, and then, and then the mouse can actually live from there on still for weeks and on after. So you can induce the injury in one kidney but not the other, and then you will have a cohort of mice that gets this nutritional supplement. We call it dicarboxylic acid either with 12 carbons or dicarboxylic acid with 8 carbon chain lengths. And that protects against the kidney injury, and it's a massive upregulation in succinylation, as you will see. And here on the right, I'm showing you, it's the nutritional supplement influences both mitochondrial modifications as well as peroxisome modifications. But we're doing everything with data-independent acquisition on the Orbitrap Eclipse, and Joanna has been acquiring all that data. So for Spectronaut, you may be familiar with these little icons on the top. You can really use this to do easy setup your method. I will show you different steps to take, defining settings for PTM level searches, to maybe having to import a protein level search into your PTM analysis pipeline, and then you may have to say, well, which files correspond in the PTM workflow versus the protein level workflow, right? Which samples correspond to each other. So I'll show you all that. And really, what I'm showing you first is how you can set up the data-independent analysis where you, with your files. It's actually very kind of convenient. You can just choose, I have modifications. If you don't choose modifications, well, you don't have to, look at the PTMs. It's a choice. We actually here choose succinylation, as I mentioned to you. So you basically just add that modification into your search engine, into Spectronaut, and and then you have like certain parameters you can set. I think we can share the slides, and also I think this is kind of recorded, so you can look back at the specific settings. I wanted to just point out a few things here. You can actually have something where you say, well, use for data filtering a Q value, and you can use imputation for the background. So in case that the PTM was not found, you make the, look in the background what the signal would have been, so you can get the quantification values more accurate. And then you can choose the run normalization here, and for in this workflow, we used to not use the cross-run normalization because we're doing PTM normalization later. But those are all quite easy things to set up, and then from there on, you can always use the methods you have defined. So it's very convenient and easy. And I just wanted to show you here also, for example, here you can actually use this normalization, what I mentioned earlier, where you can decipher protein level regulation or peptide PTM regulation. And we're using, in our case, all peptides from the same corresponding protein, so that you can do the PTM normalization for the protein regulation. So those are all choices, and the reports will give you normalized or non-normalized data down the road. In this particular workflow, we choose not to calculate stoichiometry. In the second project, we did. So here we choose no stoichiometry calculations. And, this is also the different, what I mentioned earlier to you, the dual enrichment. If you have the file where the protein lysate was separate, you would say here for the stoichiometry, you would choose modified from sample and non-modified peptides from input sample, from a different file, from a different search that you did with Spectronaut for the protein level. But if we use this single workflow where lysate and PTM was all in the same acquisition, then you can choose modified and non-modified from this sample, from this acquisition. So those two different workflows exist, and you can just choose whatever setup you had for your own sample. So we did the enrichment in this kidney project, so we chose the upper part. We call this the dual thing. So what you will have done in Spectronaut will be, these are your PTM level succinylation acquisitions. So we import those, so we kind of point to that from Spectronaut. And the next step is then you can import the SNE file, the Spectronaut file, that will be for the protein level, so because those were different acquisitions. So, you can point to that, and you can point to the protein level search that you maybe have done maybe the day before, right, or the morning before. So you point to that particular SNE file that has all your protein level results into your PTM pipeline now. And then once you've done that, you can see how they're now matched up, right? You have the PTM level, and you want to match them up, so you may have to tell Spectronaut how do they correspond, what sample is the PTM level, what sample was the protein level. So, you know, it's like a simple little Excel file where, you know, this was the PTM sample and this was the protein level sample, say, for the knockout, and this is like, say, knockout 1, knockout 2, wild type 1, wild type 2 or something, right? Or in my case here, diet replicate 1 and diet replicate 2. So it's very easy to actually set up. And then in the end, you would see both of these together here, PTM and protein level files. All right, so after, and you know, yeah, so then, you know, you can basically really see this here. Yeah, so the setup is very easy, and you know, then we can use all the normal Spectronaut features to look at retention time regression. We can use the retention time IRT peptide, and then also you can see some plots where, for example, you can already see the normal diet in red. You see less succinylation sites versus when you have the diet that I mentioned to you with the dicarboxylic acid carbon length 12 or carbon length 8 in yellow or in green. So even this very simple initial PTM kind of visualization can show you that there was a massive upregulation with this diet. Yeah, so this is, we've always liked the visualizations in Spectronaut. And just showing you here, for example, here we're looking at PTMs and we're looking at non-normalized, So, for example, here you have the no diet, the diet DC8, the diet DC12. If you do the protein level assessment, you can see that, for example, the protein level in the DC12 was a little lower, right? So this is PTM, this is protein level. So you can choose to normalize for that. And in that case, you would boost up the DC12 a little bit on the PTM level. But again, those are choices, but this can help you normalize for protein level regulation. And it can influence your differential analysis. So now, after you've done all the searches, you can actually look at the analysis post-acquisition. So a lot of machine learning has gone into the PTM analysis, doing the quantification particularly. And what is really beautiful in Spectronaut, in your original search result, you can actually look at candidate files. You can export everything into Excel, but if you just want to have a quick look, how did my data turn out, and can I just call my collaborator real quick, all the data looked great, you can look at the candidates in Spectronaut. You may be familiar with this. You can have the conditions of your quantification, and then you can even sort the file, your candidates for example, to say, only show me the succinyl. You can sort and filter for that, or you can sort for, like, say, Q value, show me the best Q values, the best confidence, or show me the highest level regulation. So you can do all that. On the fly, you can realize, oh, I really find this particular protein interesting, this particular protein group here. If you click on it, Spectronaut will just generate some, you know, kind of quick visualization here, where you can see, oh, this is how this particular PTM site looked in the normal no diet. This is how it looked with the diet and with the one diet and the other. And please, for you to see, this is a log scale. So again, there's a massive upregulation here. And you can see the individual biological replicates. So you can very quickly get a nice impression on your data and quality and what might be your most interesting PTM sites. And again, I want to point out, this is for each PTM you can visualize like this or export also later. It actually automatically exports. You can also, with this PTM analysis, do clustering. In this case, I'm showing you here normal diet injured, DC12 diet injured, and DC8 diet injured. So, and then these were the uninjured kidney samples here. So you can appreciate the clustering is day and night. Without the diet, it's very far away from either the one diet or the other. But you can also see, even though it's just the carbon chain lengths that changed here, they cluster separately. But what we were most interested in, well, what changes? How do the changes look? And you can look at that real quick. You can just click here on the volcano plot icon, and then you will see that, for example, when you have the diet, dicarboxylic acid 8 in the injured kidney over no diet injured, you can see a massive regulation of the PTM space. These are all the succinylation sites. We enriched for those. But again, I would like to point out, here you have log 10, you have log 20, and there's many, many sites that go up and also sites that go down. So somehow, just by having this diet for these mice, and this, again, I was saying the diet is protective against kidney injury, you get a massive change in your PTM portfolio. But once you take the diet away from the mice, this will all normalize again. I mean, that's the beauty with PTMs. They're so dynamic. You can have the diet maybe for somebody who will undergo cancer treatment, and you'll have the diet for them maybe before they go on the cancer treatment. This will protect their kidney, and then after the cancer treatment is finished, you can take the diet off again. This is the vision we have for the future, and these are natural products, so we hope we can do this. All right, I just wanted to show you can also do on-the-fly pathway analysis, go enrichments. In our case, we saw a massive regulation of these PTMs with the mitochondria and also in the peroxisomes and also in the complexes of the electron transport chain. And again, everything is so easy to see in these reports. You can customize your report. You can see multiplicity, how many PTMs are there per peptide. You can look at the site probabilities. That's a really important feature in SpectroNaut. You can filter by that. You can look at the non-normalized quantities. You can look at the protein normalized quantity and the normalization factor. So this is all customizable. And once you've made your report template, you can use it from there on. And there's also defaults there. And I just wanted, and of course you can also calculate the stoichiometry. Just very briefly, I keep on telling you how this is a protective mechanism for the kidneys with this diet. Indeed, this shows you here on the left an injured kidney. This is regular diet, no diet basically. On the left, you can appreciate how the kidney is damaged, right? The whole papilla in the middle is depleted with this obstruction in the kidney. You just had a little obstruction of blood flow, versus on the right, you can see a normal kidney. I mean, even me as an analytical chemist can see that the kidney on the left doesn't look healthy. When you just put them on a diet for just a week or two, it doesn't happen, right? You can see that the diet protects against this massive kidney loss that you see on the top. You can see here that with this little diet, it's basically looking totally normal, just as the non-obstructed diet. Very rarely do we see such strong phenotypes. So this is why we're so excited about this project. The succinylation protects against kidney injury. This is the PTM level effect. This is not a protein level effect. So I want to just spend 2-3 minutes on my last bone proteomics work. Why are we interested in that? Well, with aging, we have massive changes in the musculoskeletal system. This little image on the left is supposed to show a joint, say like a knee joint. You can appreciate that for example, meniscus and the synovial space is really damaged with age. And we really want to understand that better. We want to maintain bone quality. So we've developed some workflows for bone proteomics. There is also the thought that bone health, as it declines with age informs brain health, and the other way around. This is not something we think of normally, but it is true that bone health can influence brain health and the other way around. Alzheimer's and osteoarthritis are often linked and are risk factors for each other. We had some mouse models where we looked at ApoE, ApoE3, ApoE4, ApoE2. Most people have ApoE3. Those are just mutations in the ApoE proteins, very few mutations. ApoE4 is higher risk factor for Alzheimer's. So the questions that we asked here, and this is work from Cassie Liu and my postdoc Charlie Schumann, so they were asking, well, are the ApoE4 bones different than ApoE3 bones? We focus so much on the brain in Alzheimer's. But one of my collaborators, she actually would say, well, Alzheimer's is a bone disease. It's not only a brain disease. The bone quality is highly impacted in Alzheimer's patients and Alzheimer model animals. So we're looking at complicated folding of collagens, folding and three-dimensional structures. Collagens are a big part of the extracellular matrix and the matrix in bone and other organs, but in bone even more so. It gives the stability of the bone, and the cross-linking of these collagen fibers can be very complex. And this happens via hydroxyproline modifications. It's an enzymatic process via these enzymes P4 and P3, and if this hydroxyprolination doesn't happen, the entire cross-linking and bone stability is affected. So that's why we're interested in hydroxyproline. So how do we look at that in the bone? Here, I was telling you earlier, it's so abundant, this hydroxyproline, we don't even have to enrich. So we can just say the modified and non-modified peptides are all in the same peptide. So really easy setup. Once you get your results, massive upregulation of the hydroxyprolination when you compare E4 versus E3. So this is, we were hoping for some changes. What we saw were massive changes. So we were really happy about that. So, Jonna and Cassie looked closer into that. So for example, here you have a hydroxyprolination. You can extract the XIC, and actually you can see that this particular peptide either has hydroxyprolination in this site here, or in the other side of the proline that's here. So the site differentiation in SpectroNaut is really important. You can appreciate the non-modified peptide is barely there. So everything is hydroxyprolinated. Indeed, with SpectroNaut, we were able to calculate the site occupancy or stoichiometry to 84%. So what that means is all the unmodified peptide is now modified. So there was a peptide, it had a proline, everything is hydroxyprolinated. But what is interesting is also when you look at the other proline in that same peptide, just a few sites apart, it has also a really nice XIC. You can look and extract the site-defining fragment ions and extract those for quantification. And you can see that in that case, the stoichiometry and quantification is different. The stoichiometry is 65%. Honestly, still super high. A lot of phosphopeptides, for example, phosphotyrosines, are sometimes only 0.1%, and they still impact the biology greatly. So you can imagine how such a massive PTM regulation really can influence your bone stability in this case. And yeah, we get all these stoichiometry readings for free now. I mean, they're all there, and SpectroNaut can extract them out for us. Just one very brief showing you here, we can actually see that in the E3, the modification stoichiometry is around 60%, and it's quite much higher in the E4 that has the higher risk factor for Alzheimer's, and even in the E2, it's higher. So these are just things we're starting out with, but I hope I was able to show you the many new features that we now have to really dive deep into PTM analysis. And I would like to thank my lab. This was at our Christmas party. And we also have postdoctoral fellow positions open, so find me, and thank you again for giving me the opportunity to speak. Thank you.